Okay, so first let's share my review screen. Okay, so y'all have this review. We went over it for chapter 4. So here I've actually changed or added a few other points that we'll discuss today. So yours may look a little different. I just kind of reworded some stuff. I didn't really add anything new. I also added what topics were on your assignment that you're already pretty well versed in and that you know some more information about. So yeah, I took a picture of this on my phone, so I'm just gonna read kind of off my phone, but I'm mainly going to use the PowerPoint to go over these main topics. So let me share a different screen and let's get started looking at the PowerPoint. Okay, so in the nervous system, first thing that was on the review was the functions of the nervous system. So this is just the main functions which are listed on this slide. When I went over this in chapter 5, the actual video lectures, I went into detail over those bullet points there. So what you primarily need to know for the exam is this is the most important system in the body. So nervous system, most important system, primary system of communication. So this is how basically our body functions. You can see everything else that this system is responsible for, and we'll go into more detail about that. But from this slide right here, just primary system of communication, most important system in the body, and we'll touch on all those other topics in just a little bit. Okay, whoops, I don't know what I just did. So next slide, talking about homeostasis. So homeostasis and maintaining an internal equilibrium, we discussed this, or y'all discussed this on your assignment. So the first question on your chapter 5 assignment was talking about during exercise, homeostasis gets disrupted and equilibrium gets disrupted. But we want to make sure that we can get back to kind of our set point where we started before exercise. And y'all went into a lot of detail about how exercise changes basic homeostasis and how our feedback loops can get us back there. So what you'll kind of see on the exam is maybe just a point or a topic about what is homeostasis or what is equilibrium and what happens to the body during exercise, those two, which it gets disrupted. And then also just knowing the difference between a positive feedback loop and a negative feedback loop. So we know a positive feedback loop, positive means promote something or intensify something. We gave 2 examples during the lecture, blood clotting, childbirth. But what's actually more common is the negative feedback loops. So we actually see these more so during exercise. And my examples were temperature regulation, fasting, blood glucose regulation, and there are many more. So what you need to know for the exam is first just the difference definition-wise, and then probably a practical example. So if I'm exercising and this happens, my temperature increases or my blood glucose level drops, how is a feedback loop going to get me back to equilibrium? So what is my body going to do to reverse that process? So it's kind of a question that you'll see, more of a practical question on the exam. So next we get into components of neurons. So this slide right here with the unipolar, bipolar, and multipolar, you actually don't have to know the difference between those. You won't see anything like this on the exam. What you will see is your basic components of the neuron, which you may have learned in your A&P1 or A&P2, anatomy and physiology. So dendrite, cell body, or soma, and the axon. So those are going to just be the 3 primary parts. Another part you do have to know is the axon hillock, which we did discuss the axon hillock, where that's where the stimuli accumulates before that action potential is sent down the axon. So axon hillock is another component that you do have to know about. But yeah, it's going to be just kind of those components. What are they? And a good slide to look at for understanding that in more detail is this slide right here. So knowing what the motor neuron is, and then knowing all the components. Some other components you may see on the exam is myelin sheath, and then node of Ranvier. So what you see right there. So myelin sheath, we know, and we'll talk about that on this slide here. We know these are created by Schwann cells. We know the two different types of communication that neurons have based on their myelination. If you remember, think back to the case study that y'all did, and if you got the case study correct, the case study was an acoustic neuroma tumor, also called a schwannoma. So a schwannoma is due to overproduction of myelin. So that's kind of why that case study was relevant to this lecture, is because that tumor in the temporal lobe of the brain was caused by the myelin sheath is being overproduced by the Schwann cells. And so the Schwann cells were malfunctioning. So pretty interesting on how we can bring that kind of full circle back to the lecture. So yeah, it's just knowing those basic components of the neuron. Next is neuroglia. So they're glia cells. We have microglia and macroglia, which we learned about in that YouTube video. So you may not have a question specifically about what is an astrocyte or an oligodendrocyte, but you may have questions like, what's the purpose of neuroglia? So what do they do? And these 3 bullet points right here are going to be the best place for you to learn that. So just knowing that they don't conduct electrical signals, they aren't part of the communication process. They provide support, nourishment to the neurons, and most importantly, they form that myelin sheath for the motor neuron, so the axon of that neuron. So yeah, that's just kind of what you have to know about neuroglia. So next we get into types of neurons, sensory versus motor. So this one's pretty important to understand, knowing the difference between an afferent neuron and an efferent neuron. And so one way to remember that, an afferent neuron, that's going to be a sensory neuron, and an efferent neuron, that's a motor neuron. So the way I remember this in college, it's pretty silly. I may have told y'all this in the lecture. I don't know if I did. But sensory, so S, and then that is also an afferent, which is an A. So my best friend in high school was Shelby Artemovich, so SA. And she was a very strong person, a very powerful person. And sensory, we always know that sensory kind of detects the external environment. So I made that mnemonic for me in college. SA, sensory, and afferent. And then EM, or ME, I had another friend who was named Emily, and she was very shy and weak, and she didn't really do as much kind of on, I don't know, on the outside. She was more internal person. So with this motor neuron, she is processing that information and sending it out in an action, kind of in a way. So that was my mnemonic. That's how I remembered those two. So you can come up with some way to remember sensory neuron and afferent, and then motor neuron and efferent. So whatever is easy for you to remember. So you do have to know those neurons there. You have to know the purpose of a sensory, the purpose of a motor. You also have to know the interneuron. So that interneuron we know is that connector neuron. So that connector neuron is going to connect the two. It's going to fine tune the signal, and it kind of makes the signal more accurate. We know interneurons are really important in sports. They're really important in detecting external stimuli in an environment that is moving at a fast pace. So yeah, you have to know the difference between those neurons. And then next we get into 2 terms here, irritability versus conductivity. If I were you, I would just know those 2 definitions. So know the definition of irritability versus conductivity. So pretty simple. Where it gets a little trickier is understanding how impulses are actually conducted. And that's where we get into action potentials. And you did talk about this on your assignment. So you wrote about this. Some people may have not written as much as I expected them to. So here we can kind of talk about what are the most important points about understanding the action potential. So first right here, we have to understand that at rest, so when a neuron is not active, when there's no movement, no activity, no signals being conducted, that neuron has a negative charge. So when no activity is going on. We also know that when we are not at rest, that charge is going to change. It's going to become more positive. And we'll look at that in just a second when we talk about the role of sodium and potassium. This here just talks about permeability. So what is permeability? The ability of ions to move across a plasma membrane, difference in ion concentrations. So this is where we get into concentration gradients and how that also helps the action potential to be conducted or the impulse to be sent. And then the sodium and potassium pump. So we'll get to that as we move through this. So like we said, resting membrane potential, negative charge on the inside of that neuron, and there's a positive charge on the outside of that neuron. You do have to know resting membrane potential is negative 70 millivolts. Also remember, that's just an average number. Depending on the size of the motor neuron, that's going to determine if a neuron's more negative or more positive. But that negative 70 is just like that average number that we give most motor units or motor neurons. You also have to know about reaching the threshold. And we said that the threshold is reached at the axon hillock. And so that's when the membrane potential changes. So now the membrane potential is going to reach that threshold. So it's becoming more negative. And so that's going to be negative 55 millivolts. So that's our nervous impulse. So that's how it will occur. Oh, sorry, I meant it's going to become more positive. That's what I meant. It becomes more positive. So negative 55 is higher than negative 70. It's closer to 0. So we're getting closer to that positive range. All right, and then this slide here, we talk about the channels or the sodium and potassium gates. And so this picture, looking at this one on the right side, a little tricky to understand, but you have to separate the top and the bottom. So let's just look at the top part where it says depolarization. So if you see, it says cell exterior and cell interior. So we just said there's a difference in charges between the outside and the inside, where the inside's negative, the outside's positive. So we see here that when depolarization occurs, after we've reached that threshold, that the sodium channels will open, and then sodium will enter into the cell because it was on the outside of the neuron. Now it's going to move to the inside due to those gates. And that's when the membrane potential changes and becomes more positive. We get up to positive 30. Okay, so now we're at positive 30. Now we're going to go back towards that repolarization, or in this case, it's going to be called hyperpolarization. And now in that process, the potassium channels will open, and then potassium will leave the cell and move to the outside. So they basically just switch. But once the impulse is conducted, we have to return to resting membrane potential. How do we do that? The sodium and potassium pump. So we see down in the bottom corner, for every 3 sodium that are pumped out, it's going to pump 2 potassiums back in. And so that's what that picture shows. It's just this pump that just moves those ions back to where they're supposed to be, which gets us to resting membrane potential. And this here just kind of puts it all together. So when we look at this slide, we have our numbers and we can kind of follow what's happening. So like it said, we start at resting membrane potential, millivolts negative 70. So we have a stimulus to depolarize. So we've depolarized the action potential because that threshold was reached. Sodium channels open, they enter the cell because sodium's on the outside, and then potassium will move to the outside because we know it's on the inside, so it's going to leave the cell. And so as we follow those numbers, you can just kind of follow what's going on. Before we return to resting membrane potential, we're going to hyperpolarize, which we see that red arrow down there at the bottom. So that's kind of basically one action potential, which I went through very quickly. So if you need more help understanding this process, return back to chapter 5 video lecture so you can hear me go over it in more detail, or watch those videos that are posted, which are really great videos that explain this. But what we want to understand is what's the big picture? So how do we put this together and make it more practical or make it easier to understand? So we talk about this now with two neurons connecting together. And so that is synaptic transmission. So you wrote on your assignment, let me go back and look, about synaptic transmission, which is also communication across the synapse. So that was a question on your assignment. So you should know a little bit about this, but let's go over this again. So synaptic transmission, we have our, let me make that smaller. We have two neurons connected together. And so what we just went over with the action potential, that's happening down the axon. So the axon of a motor neuron, when we talked about that action potential, that process is occurring all the way until it reaches the end of the axon, the terminal buttons. Those terminal buttons are going to be connected to another neuron, or they can be connected to muscle fibers, which would be called a neuromuscular junction. So what you want to understand, synaptic transmission is just the communication of neurons. The synapse is the gap. And so we're going to talk about how that process happens basically when two neurons are connected together. So you want to think, okay, we just had the action potential. The action potential was sent down the neuron. So it's sent down the neuron. Sodium and potassium are being passed back and forth as the action potential hops or leaps into those nodes of Ranvier. So the myelin sheath, we said, is going to speed up that electrical conduction. So now that action potential just hops and leaps all the way across the axon until it reaches the very end. Okay, and now this slide right here, we're at the very end. So now that action potential that just traveled down the axon has arrived at the presynaptic terminal. So that's those terminal buttons. Now that presynaptic neuron is going to release a chemical, which is a neurotransmitter, and it's acetylcholine. Acetylcholine is the chemical that's released. It's going to diffuse across the gap, and it's going to activate the receptors on the postsynaptic cell, and it's going to bind to it. So hormones, we'll learn this in chapter 8, but hormones are going to bind to a specific receptor that it's designed for. And when it binds to that receptor, it's going to allow the opening of more channels, the opening of gates, and it allows that action potential to now enter into the postsynaptic cell. So we're in the other neuron. So it's going to cause depolarization of that second membrane, the postsynaptic membrane, and all the ions can move across. So then the action potential will be processed by the postsynaptic neuron. It's going to be processed in that soma and then sent down the next axon. So that is synaptic transmission, and this picture kind of shows that, and the videos at the bottom are really good if you need to watch those again. But if you look at this picture, we see the nervous impulse arriving at the terminal buttons, which is the action potential. We see these synaptic vesicles. They're going to release the neurotransmitter, which is acetylcholine. Calcium diffuses into the terminal buttons, also plays a role in this process, and then it diffuses into the other postsynaptic cell, and sodium and potassium enter into it, and there we go. We have the passing of that action potential to the next neuron. So you wrote about that on your assignment, so should be pretty familiar with that process. But that concludes part one. So now we're moving on to part 2, which gets into more divisions of the nervous system. So we're now just going to focus on the brain for a little bit, and then we'll move back and we'll talk a little bit more about that action potential and how it's going to activate the muscle fibers. And we'll get into that at the end of this lecture. So what do you need to know for the exam? You need to know just the difference in those 4 lobes. What does each lobe control? So you may have a question like, what does the temporal lobe control? You should know about the temporal lobe, since that's what our case study was about. It was about a malfunction to one of the chromosomes that take place in that temporal lobe. And then cerebellum, corpus callosum, just kind of knowing what each of those structures are. This here just shows you where those are located in the brain. You do have to know that the motor cortex is in the frontal lobe. So Tyler, you go get her. Motor cortex is in frontal lobe. And what else? You don't really have to know anything about Broca's area or Wernicke's area, just kind of the basics of each lobe. You also don't have to know about the cranial nerves. Here was just a neat study that looked at motor cortex activity during exercise. You don't really have to know this for the exam. It's just neat to understand how exercise allows activation of the brain. This slide you do have to know. So this slide, the diencephalon, the hypothalamus is a really important concept to understand because we're going to talk more about the hypothalamus in chapter 8 when we get into hormones, because the hypothalamus has a lot of control over hormone regulation. So in the lecture, I told you what the hypothalamus regulates, like thirst and temperature and endocrine function, which is hormones. So you do have to be familiar with hypothalamus, diencephalon. You have to know the brainstem too. So what is the brainstem control? I also went over this in the lecture. And the limbic system, just know it circles the top of the brainstem. It works with, I think, conscious control. What's the word I'm looking for? Just other kind of thoughts and processes that we have, emotions, sensations that we're feeling, thinking. But yeah, so just knowing that those structures of the nervous system. Another one you have to know is about the spinal cord. So the main function of the spinal cord is to regulate our reflexes. So reflexes when it comes to exercise, or sports, so how quickly we can react to something, or if we touch something hot and have to remove our hand. So all of our reflexes controlled by the spinal cord. So you kind of have to be familiar with that, control sensory operations, motor activities. This slide here just shows how it innervates different muscles, what parts of the spinal cord or the spinal nerves innervate which parts of the body. You don't really have to know that part, just the main purpose of the spinal cord. And where does it say that? It says it right here. So spinal cord is the site of our reflex actions. Knowing that it is important in sports, we talked about how that the more trained you are, so higher functioning athletes, they don't have to send signals to their brain. They can process them more centrally at the spinal cord. So that's a really positive adaptation. It means that that really talented athletes, they have faster reactions. So they can react really quickly due to the decreased amount of time it takes to just send the information to the spinal cord and not have to send it all the way to the brain. So yeah, just another benefit of that. Next, kind of more looking at this, or still kind of looking at the spinal cord, we went over this reflex, the simplest reflex. We talked about how the sensory motor neuron is sent to the spinal cord, processed by the interneuron, sent back with a motor neuron. So yeah, how this process works. So do kind of be familiar with the myotatic reflex. Know it's the simplest reflex and it's monosynaptic, means there's only one synapse. Next, let me look at the review. So it does say that reciprocal innervation is on your exam. Going over this, it's pretty simple to understand. It's easier to understand when you apply like an example to it, which we can do. So looking here, we see that, and it's basically, well, it's this slide right here and the last slide too. But it's when the afferent nerve synapses with the inhibitor, but also with the initiator, but also with the inhibitor. The way you want to think about it is doing a leg extension, just like in the picture. If the goal is to activate the quadriceps during a leg extension, we don't want to activate the hamstring. And so during the process of quadricep activation, there's also hamstring inhibition, where it's not activating the antagonist, which is a positive adaptation. And so just knowing what reciprocal innervation is, it's when the neurons can activate the agonist muscle and shut off the antagonist. It allows for greater force production and more training adaptations. Okay, so next let's move on to the peripheral nervous system. So peripheral nervous system first, let's talk about autonomic. So we know autonomic is going to control everything that we can't control consciously. So everything that's unconscious. So such as heart rate, blood pressure, breathing, digestion, and reflexes. Even though breathing we do have a little bit of control over, but eventually it will be taken over by the autonomic system. We know the autonomic system has two branches, so parasympathetic and sympathetic. So first, talking about sympathetic. So sympathetic, we know what part of the brain is activated during the sympathetic nervous system stimulation, the amygdala. You do have to know that. So remember, the amygdala is something you have to know about on the exam and know it controls some of that sympathetic stimulation. You wrote about this on your assignment, so you're probably pretty familiar with sympathetic nervous system. You do have to know the hormones here. You have to know that epinephrine is secreted in high amounts when our sympathetic system is activated. We did say, though, that norepinephrine is active, but it is released in very low levels throughout the entire day. What does that allow for? It keeps us alert. It keeps us awake during the day. But epinephrine, a huge surge of it is sent into our system when our sympathetic system is activated. What does it do? Increases heart rate, blood pressure, blood flow, releases glucose from the liver to give us more fuel, to give us energy. Same with the breakdown of muscle glycogen. More things happen during this stimulation, but these are kind of the most important ones that you need to know. We will talk more about epinephrine in chapter 8 because it is a very important hormone. Next is the parasympathetic system. This system releases acetylcholine. You have to know that. It helps us get back to homeostasis, back to equilibrium. So when stimulated, you have to think everything that returns us back to a state of calm. So like after exercise, we're trying to recover. After, getting really upset or crying, we have to calm down. So this system is going to be active in those examples. All right, next, moving on to the peripheral nervous system. So now we're in sensory somatic. So when I pull this slide up, you'll be like, oh my gosh, we just talked about sensory and motor neurons. Yes. So everything we talked about earlier with synaptic transmission, how we have a sensory neuron that's going to initiate the motor neuron, well, that's the sensory somatic system. So what we explained earlier is the same thing. And we are also said it uses acetylcholine. That's the neurotransmitter that's going to be passed across the synapse. So it's going to move across the gap. And that's what we see in this picture. This picture, though, looks at the the motor neuron is activating, is activating the it's at the neuromuscular junction. It's activating a muscle fiber. So activating this muscle fiber right here, whoops, I didn't mean to move that. Yeah, so sorry, I'm a little distracted with my family here. So using acetylcholine to pass across the gap, and that's how we have muscle fiber activation. We're going to get to muscle fiber activation in just a little bit, and when we talk about motor unit recruitment and rate coding. But let me kind of finish up this part here. This slide, like I said, is a really good studying tool because I've kind of separated all the divisions and kind of everything that you have to know for this. So a good studying tool, just kind of looking at this slide, separating everything. You'll have probably about 4 or 5 questions about, peripheral nervous system, autonomic nervous system, and all the divisions. So be prepared for lots of questions about those systems. All right, so we've made it to part 3. So this is going to be the last part of this lecture, and we're going to tie back in what we learned earlier about synaptic transmission and now activating muscle fibers. So let's bring everything we learned full circle and start back with the action potential. So, or we can even start with, building up a threshold of stimuli. And when we have enough threshold of stimuli, when we reach that threshold level, the action potential is sent. when it's sent down the neuron, it's going to activate a group of muscle fibers. And so that's how we have muscle contraction. And so here, what this goes over, we know that motor neurons, motor units contain the neuron and the fibers. So when we talk about a motor unit, let's pull out that green motor unit right there. We see the neuron that came from the spinal cord and it attached to the muscle. So it's a pretty long distance. Well, that entire structure, that's the motor unit. It's the neuron and the fibers. You do have to know that for the exam. Neuromuscular junction is the connection. It's like the synapse point, but instead of connecting to another neuron, we're connecting to the muscle fibers, and that's the neuromuscular junction. You also have to know that. All right, here we talked about fiber type. In chapter 4, we learned all about fiber type. We learned a little bit about how the muscle is structured, how it's put together. But here we want to understand how we activate different groups of motor units or motor neurons. And I went over this pretty well before, talking about how each of the 1, 2, and 3 motor units, they're different. They contain different fibers that have different characteristics. So maybe slow, some may be fast, some large, some small. So just kind of remember that. The motor unit is the neuron and the fibers, and it's controlled by the spinal cord, or the neuron in the spinal cord. So let's talk about this part right here. You may have a few questions about threshold level. And so when we say that type 1 slow twitch fibers have a lower threshold, that means they're easier to activate. That means that normal day-to-day activities, the threshold is not very high. It's pretty low, meaning it's easy to recruit those. Versus a type 2 fast twitch has a higher threshold, meaning it's more negative and it takes more stimuli to recruit them. That's why they're usually only activated if we're lifting something really heavy, or running really fast, or jumping, or moving our body really rapidly. It takes a lot of effort to recruit the type 2s. So that's kind of how you have to remember that. Difference in number of fibers per motor unit. We said that was called the innervation ratio, which I have right here, example of an innervation ratio, saying that the muscle or the motor units, let's say in a large muscle like our quadriceps, have more fibers per motor unit, where the motor units, let's say in my hand, have very small number of fibers that are connected to that neuron. So that's the innervation ratio. It's the difference in, or the number of fibers in the motor unit. So we have larger motor units in larger muscles, smaller motor units in smaller muscles. So makes some sense there. Next, here we're getting into the size principle of recruitment, which you did write about on your assignment. So you should know about this. It has to do with the effort, like we said, how much stimuli is present in order to activate certain fibers. So we see here recruitment of motor units accomplished by varying the strength of the electrical stimulus. So the more stimulus, the more fibers we can activate. So this down here just looks at the proportion of motor units in a muscle that's activated. So saying that if we reach the threshold, we're going to activate some fibers, but if we have more stimuli sent to the muscle, we're going to activate more fibers. This next slide continues, and this is where we gave the practical application of motor unit recruitment. How if I'm lifting really light weights for a high number of repetitions, I'm only using my type 1 fibers because the task at hand is not very hard, and my type 1 fibers, that's all I need to help me lift that weight. Where when the weight gets heavier and the repetitions get lower, I have to recruit larger motor units or larger muscle fibers to do the job. So I have to recruit my type 2 fibers along with my type 1 fibers to help me do harder tasks. So y'all probably understand that concept a little bit better after doing it on your assignment. But this part here, let's get into all or nothing. There's one question on the assignment about the all or nothing law or all or nothing principle. And this has to do with motor unit activation, again, determined by that threshold, which is something that we've been hearing throughout this entire lecture. So basically saying if we don't reach the threshold, no muscle fibers in that motor unit are active. So let's take just like a muscle fiber in my, or a motor unit in my bicep. So motor unit in my bicep, let's say it has, 20 fibers attached to that neuron that came from the spinal cord that's activating it. Well, if the motor neuron itself, if it didn't have enough stimuli to send an action potential to the muscle, then no fibers are going to be activated. So none of the 20 fibers in that one motor unit will contract. But if that threshold is reached and that action potential is sent to my bicep, then all 20 of those fibers connected to that neuron, they will all activate together. So if the neuron is sent, however many fibers is connected to that neuron, all those fibers will activate together. Something you have to understand is if one motor unit in my bicep is activated, it doesn't mean the motor unit right next to it is activated. So motor unit activation is just, it depends on how much force I'm trying to, produce, or how much activation of the entire muscle I have. So just because one motor unit is active doesn't mean a motor unit next to it is also active. So that's kind of one way to think about it. And let's continue that topic with motor or multiple motor unit summation. So how do we produce more force in the muscle? If one motor unit is active in the muscle, just like I gave an example of, that's not going to produce a lot of force. So if one motor unit in the bicep is active, I may not be able to do very much with just those fibers that contracted. But more motor units that are active in a muscle, the more force I can produce. So that's where the force comes from. It comes from activating more motor units, having more muscle fibers contract. That equals more force. Wave summation is another way to increase force production, and we have an example at the bottom down here. So one single twitch, so we see with stimulus and we see a twitch. So that's just like a twitch in the muscle. So like one little tiny activation. Again, very little force is produced with a twitch. You may have to know that too. A twitch produces the lowest amount of force. Wave summation will produce more force because you see over here, the tension's getting higher because the muscle has less time to relax. So if I'm contracting a muscle, and then before it completely relaxes, another stimulus is sent. So that means the tension's going to keep increasing and increasing. So wave summation, or gradation of force, that's like a stair stepper in force production. So that's another way to increase tension. How do we do that? How do we increase tension, or how do we create wave summation? By rate coding. So rate coding is controlling the rate at which the action potentials are sent to the muscle. So when we see the picture of wave summation, we see those arrows, those are action potentials. So as the action potentials are being sent, they're being sent in a shorter time frame, which means we're activating the muscle at a faster rate. So this picture shows it right here. We have an action potential. One action potential causes a single twitch in the muscle. Again, very low amount of force, not much activity. But if we send two action potentials together, we have wave summation. So we have increased that force because the muscle can't completely relax yet. So rate coding is also shown in this picture right here, or it's also called manipulation of firing rate. And we see the arrows are being sent to the muscle rapidly and rapidly and rapidly with very little time in between. Look what happens to that tension. That tension increases, that force production gets higher and higher. That's called tetanus. You do have to know this for the exam. Tetanus is the most amount of force that can be produced in the muscle. It's the strongest contraction. It means the muscle cannot relax, and there's just too much stimulus going. A practical example is like a cramp. When we cramp that muscle, or that muscle cramps up on its own and it doesn't relax, that is an example of tetanus. Another example is like if we're doing an isometric contraction and we're holding a muscle in a contraction for a long period of time, that's another example of tetanus, except for all the muscle fibers, have to be active to create that much force. So yeah, you do have to know the difference in a single twitch, wave summation, and tetanus. All right, neural drive. So neural drive is a training adaptation, and we're in adaptations. We're kind of towards the end of the lecture, almost done.
Yeah, that's the best way to go here.
Okay, neural adaptation. So neural drive is basically like combination of we have lots of motor units that are recruited, but we're also rate coding those motor units. When we do that, it allows for more force. So when we hear that someone has an increased neural drive, it means it's voluntary activation of motor units and they're stronger. So adaptations, number one, neural drive. If we have more neural drive, we have greater force production. And it's due to we've recruited more motor units and we keep stimulating those motor units to produce force via rate coding. So other adaptations, an obvious one, increased strength. So we're stronger due to the increase in neural drive. Increased motor unit firing rate, which is also called rate coding, which we just said. We have motor unit recruitment, we have rate coding, we have increased neural drive. Synchronization of firing. This means that we're increasing the likelihood that multiple motor units will fire together. This actually doesn't increase force, but it increases power production. We're more powerful, when we have synchronization of firing. Again, increasing the likelihood that multiple motor units will fire together. And then we also have increased contractile response to submaximal neural drive. So this is just like submaximal meaning non-maximal. So let's say we're 50-60% of our 1RM or our maximal effort. So that would be submaximal neural drive. Again, meaning that we're stronger when we're submaximally working as well as maximally working. All right, moving on, you do have to, whoops, don't cut that. No, sorry, this slide right here. This slide right here talks about training over time and how muscular changes and neural changes will start to overlap. So we see here that the first 4 to 6 weeks of training, we just started a new training program, we're lifting weights, we're getting stronger. Why are we getting stronger in those first 4 to 6 weeks? It's due to the neural adaptations that I just mentioned on the last slide. So while those are all neural changes, the muscle hasn't started changing significantly yet, but we've had changes take place to our central nervous system, and that's why we're stronger. But after that 4 to 6 week mark, you see around 8 weeks, the changes to the muscle start becoming more significant. So when we start lifting, there are changes to the muscle. The muscle is going to probably be sore. We have maybe increased size early on, but significant changes to muscle size called hypertrophy don't occur until later on. So the 8 to the 16 week mark. So at about 16 or week 16 or so, we have significant changes to the muscle that have kind of started to plateau off. And now the size of the muscle is contributing to why we keep getting stronger. So pretty interesting. I probably went over it better in the actual lecture, but you do have to know this for the exam as well. And then last, we kind of talked about the difference in motor unit recruitment between a marathon runner and a sprinter, and how these two individuals may see different recruitment patterns or recruitment activation during their event. You also probably kind of wrote about this a little bit on your critical thinking in your assignment when I said use an example to explain the size principle. So in that example, you probably said something about aerobic exercise, how it uses type 1 fibers. It goes through asynchronous recruitment versus versus the sprinter. Did I say that right? The marathon runner, type 1, the sprinter, type 2, which goes through synchronous recruitment. So this is an application question, a critical thinking question that you may want to just kind of be familiar with, like how motor unit recruitment works in a practical setting. If I'm a sprinter, or if I'm a marathon runner, or if I lift weights, or if I cycle, what recruitment is my body, what fibers are being recruited, kind of all of that. So more practical. All right, we finished the lecture. You have a quiz today. So chapter 5 quiz review, which you probably already saw in the lecture. You will have very similar questions that you see on the quiz on the exam. Sometimes they're the same questions that I just take from the quiz and put them on the exam. So make sure you kind of know those pretty well. But yeah, that's it for this. One more thing is I didn't show these, but these are great videos. This crash course website, he does such a good job. Skeletal muscle nervous system. He does basically every system. So I've put the seconds. That's the seconds there if you haven't looked at it yet. So 2 minutes, 23 seconds of this video right here. He talks about action potentials if you're struggling with that. At this time frame right here, he talks about synaptic transmission. These videos, yeah, you get it. So I think they're really helpful. I think they help reiterate what I'm saying. You can hear it from somebody else to maybe learn it a little better. But yeah, so that is all I have for this review session. But I am going to open the floor and see if anyone has a question. Ask anything you can now for me. But yeah, I'll let you think about it and then ask me something. I'll give you just like a few seconds to think. Well, it looks like everybody is still muted. So if nobody has any questions, then we can end this session. I do thank you for being here. I would not be doing this if it wasn't for y'all sitting right here. So I really appreciate your taking the time out of your morning to do this with me. This will be recorded and put on Blackboard if you need to rewatch it. If you have any questions, please feel free to email me anytime with your questions, or we can set up a Zoom session before the exam, which is on Monday. Your quiz is today, but other than that, I hope you all have an awesome weekend. And again, thank you for being here, and I will talk to you guys soon. Okay, bye.